Okay, so today we're going to be talking about the discovery of DNA and all of the scientists that were involved in the process. So first off, we're going to start with a little bit of background on DNA. So DNA is made up of nucleotides, and it's made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base. So for DNA, the 5-carbon sugar is going to be deoxyribose, and this is in contrast to RNA's ribose. And we have 4 nitrogen bases for DNA, and these are adenine, guanine, thymine, and cytosine. Also, remember, adenine is going to pair with thymine, and guanine is going to pair with cytosine. Adenine and guanine are purines, and thymine and cytosine are pyrimidines. Okay, so first off, we're going to start with Frederick Griffith. So, Griffith discovered that genetic information can be transferred from one cell to another, and he also discovered that harmless bacteria can be transformed into harmful bacteria. So he conducted his experiments using mice, and he injected them with non-virulent and virulent strains of bacteria, and used heat to see how the mice were going to react, and this is how he came to many of his conclusions. Next, we have Rosalind Franklin and Maurice Wilkins. So they used X-ray crystallography, and this allowed them to display clear images of DNA. And using this, they were able to determine that DNA structure is based on phosphate location and the backbone. Much of the research that they did influenced Watson and Crick, and perhaps Franklin would have been recognized more for her work with a Nobel Prize if she were alive, but since she worked with so much radiation during her life, she did pass away of cancer. So right here in the bottom left, we have a picture, and this shows the picture that they were looking at, and this allowed them to determine that DNA does have a double helix shape. So next we have James Watson and Francis Crick. So they developed the double helix model. Again, most of their information did come from Rosalind Franklin and Maurice Wilkins, so many argue about whether they deserve the credit that they got. But however, they did win Nobel Prizes for their contributions, and they also discovered that it is hydrogen bonds that hold together the two DNA strands, and hydrogen bonds are one of the strongest bonds that occur in the chemical world. Next, we have Irvin Chargaff. So Irvin Chargaff discovered the base pairing rules. So he discovered that adenine binds with thymine and cytosine binds with guanine, and the ratio of adenine plus guanine to thymine plus cytosine always equals 1. Next, we have Matthew Stanley Messelson and Franklin Williams-Stahl. Okay, so they discovered that DNA is replicated in a semi-conservative way. So what this means is during DNA replication, we know that DNA helicase goes down and it unzips the DNA, and then DNA polymerase goes in and adds nucleotides. But one strand of the DNA is still going to be the one old strand, and the new bases are going to be added. So half of it is conserved. So this is the semi-conservative way because half of it is still the old strand. So next we have some review questions. Take about 30 seconds to answer these. Okay, so which of the following is not a component of a DNA nucleotide? So this answer is going to be D. Yeah. So second, which scientist developed base pairing rules? So this is going to be Erwin Chargaff. And what does adenine pair with? And this is going to be thymine.